This is the first time that I've used Canvas for basically grading all of these separate components. And I'm going to basically try and have all of the criterion that you're going to need for each of these respective things posted in Canvas shortly. So I'm hopeful we'll get that knocked out this weekend. We're going to finish up our discussion of toxins today, and we're basically going to really drill down on AB toxins. We could probably spend a lot of time on the other toxins, maybe type 1 and type 2 toxins that we've covered, but I think these are the most interesting. Mostly because they're so specific, and that they essentially are a molecular analog to what we saw for sort of the whole pathogen entering into a host cell and causing some change in host cell behavior. But these are essentially just protein packages delivered to the host cells by pathogens. But before we get going, are there any questions? The last lecture was pretty good. Okay. Well, hopefully today is even clearer. But, so I'm going to cover a number of toxins, and what I'm trying to do in today's lecture is to highlight both their structural diversity and their biochemical diversity in terms of what they do, because it is really quite extraordinary how diverse this family of toxins really is. And so backing out to our roadmap, where we are, where we've been for Tuesday's lecture and today's lecture is really this green box. So there's all of these pathogen-driven processes that are key components of their infectious life cycle. And what we're going to get to today is, and we've seen how some of these processes connect to tissue damage and spread within the host, but we're going to really delve into AB toxins today, contrasting our discussion of type 1 and type 2 toxins. And so the exam 2 is essentially this kind of package here, this idea of adhering, what happens after adherence of the intracellular, extracellular pathogen, what are pathogens doing to extract the nutrients from the host. Arguably, this is one of the most exciting areas of infectious disease research in terms of bacterial pathogenesis right now. A lot of the mechanics of how this gets done has been worked out over 30 years. And we're finally able to use some really elaborate techniques to understand what are these pathogens actually eating? What are they actually living on inside of a host? So if you know this, you can hopefully alleviate some of this, right? So if you remove the ability of a pathogen to survive in a particular environment, you can obviate some of the aspects of the disease presentation that we see during infections. So where we're going to go next is delving into some aspects of exposure, and then also into some of the evolutionary aspects of what are pathogens. When you think back to the much earlier lecture that I gave you, that there's a lot of bacteria that are naturally associated with us that are the same species as some really nasty pathogens, and understanding what drives the emergence of new pathogens or the shaping of existing pathogens. So today we're going to talk about a lot of different toxins, and this list is much longer. But the things that I want for you to see are the different chemistries that go on in terms of what these active subunits are doing, and that there are a lot of different types of toxins, and they can be exquisitely sensitive to binding to and eliciting responses from very specific host cell types. And so just to review, what is an AB toxin? There are two component toxin systems. So there's basically a binding domain and an active biochemical, enzymatically active domain of these toxins. And what they're characterized by doing is that this B, or binding subunit, facilitates binding of this toxin complex to the host cell surface. Host cells take these toxins up into vacuoles, or small endosomes, and that this process of basically phagocytosis of these toxins results in release of fragment A from the host into the host cell cytoplasm, or in some cases, we'll see it later today, into different organelles. And the net result then is that this active subunit does something to the host cell machinery. We'll see it can mess with translation. We'll see that it can mess with different signaling pathways. We'll see that some of these toxins are proteolytic, that they cleave specific signaling proteins or specific basically membrane receptors. And in some cases, there are nucleases. They basically will destroy the host cell's DNA. And we'll see what some of these effects are in coming examples. And so when we think about AB toxins, there are really two fundamental ways that they gain access to the host. And how this active subunit is delivered. So the first and foremost, just like we saw in uptake of a pathogen, like kind of a whole bacterial cell into a host cell, is that everything that comes in an endosome. Okay, so basically, some form of phagocytosis results in uptake of this. In this case, our toxin enters the cell, and here's where there's some fundamental differences in how these toxins work. Okay, so some toxins require that this endosome basically undergoes the natural course of events for endosomes. And one of these is that even if they're not fused to a lysosome, endosomes typically get acidified. And acidification of this vesicle or this small endosome results in activation of this toxin and release of the active subunit out into the cytoplasm, where it can go perform its enzymatic activity. And we'll see what some of those activities are shortly. And the other is that some vesicles, and so you can kind of, in some ways, think about this as a kind of somewhat stochastic process, that some vesicles get acidified, and some will undergo retrograde trafficking, where essentially this vesicle is delivered to the Golgi, and basically through retrograde transport, the contents of these vesicles is brought to the ER. And there are specific enzymes in endoplasmic reticulum that are required for basically cleaving and activating these toxins, essentially delivering them out into the cytoplasm. So this is a much more complicated process, right? So you're relying on a lot of host processes for this toxin to be released and go do its job, whereas this is much more kind of direct. And we can kind of draw some analogies to what we saw with pathogens and their sort of life inside of vacuoles, or whether or not they just escape the vacuole and then go on and do their dirty work in the host cytoplasm. So there's kind of some interesting parallels at both the molecular level and the cellular level in terms of what pathogens are doing to interact with host cells. So, are there any questions about these two mechanisms? So I'm intentionally staying pretty high level with this, primarily because you're not required to have taken a cell biology class today. Are there any questions?
So overall, what is the purpose of these to release the A domain into the cytoplasm?
Exactly.
Okay.
Yep. So these are sort of two roads to Rome, right? Just one is very long and complicated, and exploits one set of host cell processes to basically secrete a protein in a vesicle out into the cytoplasm. Similarly, this other route is basically a very direct kind of active mechanism of transporting this active domain payload out into the cytoplasm to perform an enzymatic task. So am I going to ask you, these very special protein names here and there? No. But the big idea is that retrograde trafficking is one route, and vacuole acidification is another. And we'll see some contrasting examples. So I think the easiest place to begin thinking about AV toxins is diphtheria toxin, in part because it's structurally pretty simple, and it illustrates a lot of points about this first pathway for AV toxin activity, what's going on. So a little bit of background. Diphtheria toxin is produced by a bacterium, Corynebacterium diphtheriae. It's the causative agent of diphtheria. And so it's a gram-positive rod. And so I talked a little bit about diphtheria before, and that it is a very deadly disease, especially for children. It's currently controlled by vaccination, but due to lax vaccination implementation in various places and sort of relaxed requirements for vaccination, we're actually seeing basically reemergence of otherwise very well controlled infectious diseases. And so, like we've seen before, that production of villi are really important for colonization of the throat. And so that was kind of where I introduced this guy to you before. And so it's, again, characterized by formation of this pseudomembrane at the back of the throat. It's basically just a really big, nasty biofilm that once these Corynebacterium diphtheria have colonized, they begin secreting a toxin, which elicits more systemic responses from us in terms of hurting us. So one of the things that happens because of this toxin is it basically paralyzes ciliated epithelial cells in your throat. And so you're basically no longer able to expel these bacteria in this biofilm. And eventually this will kind of cover your throat and you suffocate. So it's a really, really terrible thing. Primarily affects children. And is kind of this reemerging, an example of a reemerging pathogen. We'll talk about some of those concepts a little bit later. So, diphtheria toxin, it's produced by bacteria when they're colonizing the throat, but because of its proximity to your throat and basically highly vascularized epithelial tissue, so if you've ever kind of bit your cheek or bit your tongue and just bleed like crazy, or you've scratched your throat, whatever the case may be, that it's very soft, thin mucosal epithelial tissue, and is really integrally connected with our vascular system. And so when these bacteria in your esophagus and nasopharynx start producing this toxin, it rapidly gets into your blood, and you can end up with essentially this exotoxin in your blood, and then a systemic response to this toxin. Beyond the symptoms, so what is this actual toxin? So I gave you some arrangements of toxins last time, that they can be a single chain, they can be separate subunits, they can be kind of a multimeric kind of heterodimeric complex, and we'll see examples. And the examples I've chosen, I'm trying to highlight both their physical arrangement as well as their enzymatic activity, and that they do very diverse things. So, what does this particular toxin do? It binds to a particular epidermal growth factor precursor that's present in a lot of basically epithelial cells, and so this particular toxin is very good at interacting with epithelial cells, but will interact with a lot of other tissues that also produce the same this host cell receptor, surface receptor. So it's delivered to the host cell cytoplasm via endosome acidification. So if we go back to our chart of these two roads to Rome, this is through endosome acidification, and the chemistry that performs is it ADP ribosylates elongation factor 2. So elongation factor 2, as I'll show you in a second, is responsible for helping charged tRNAs enter the ribosome and facilitates basically faithful charged amino acid loading into the ribosome and basically protein elongation. And so what we're going to see is that if you chemically functionalize that protein, you're essentially adding on a big molecular, well, I shouldn't say big, but it's a fairly large secondary kind of secondary compound that's being added on to an enzyme or added on to a structural protein that is going to essentially inhibit this protein's function. And we'll kind of really dig into what that looks like. So, as I said, diphtheria toxin, the active subunit gets into the host cytosol via endosomal acidification. So, diphtheria toxin is it's a single chain AB toxin. So there's one active domain and one binding domain in this protein. And if we look structurally, there's also this other kind of domain in here that basically this whole thing here we can kind of think of as this receptor binding B domain, and then this A active site domain up here. So this binds to that growth factor that's on epithelial cells and results in uptake in a small vesicle. This vesicle gets acidified, as is what happens with most vesicles that get taken up from the extracellular environment. And this induces a conformational change in essentially autohydrolysis of this A subunit from the binding subunit. And the net result is that this B subunit also facilitates egress or exit of this active domain into the host cytoplasm. So once in the cytoplasm, where are a couple of places that you would find ribosomes in a eukaryotic cell?
Cytoplasm and ER.
Okay, yep. So the cytoplasm and the endoplasmic reticulum. And so, and both of those require elongation factor 2. And so elongation factor 2 is a highly abundant protein that basically has transient association with the ribosome. And what this active domain does is it takes an NAD, a molecule of NAD, and uses that and essentially transfers that NAD onto a histidine residue of elongation factor 2, adding on ADP ribose. Okay? And what that does is essentially inactivates EF2. If EF2 is inactive, protein translation is inhibited. So why, as I mentioned before, is that elongation factor 2 is one of the components required for faithful charging or loading of the ribosome with charged tRNAs. And basically allows for the protein to continue to elongate with new charged tRNAs coming in, and the ribosome kind of ratcheting down mRNA translating the appropriate protein. So what the active domain of diphtheria toxin does, ADP ribosylates elongation factor 2. And so now the essentially binding pocket for tRNAs in elongation factor 2 is no longer accessible to tRNAs. And so essentially these tRNAs can show up and they can interact with the ribosome, but that interaction is never stabilized because elongation factor 2 is inactive. So what happens then is that when a sufficiently large amount of elongation factor 2 has been ADP ribosylated, essentially protein synthesis stops because all of the elongation factor 2 that's been produced by these cells is inactivated. And so you might say, well, how does this happen with such a small amount of toxin? Most of these toxins that we're going to see, these active subunits, are really highly catalytically active. So they're really, really fast. So their turnover rates, or they're like sort of Kcat or Km, for performing these reactions is really, really, really fast. So they rapidly convert EF2 to inactive form. This is an irreversible reaction. And so this EF2 is dead. And so once you've eliminated all of the EF2 in the cell, you can't make any new EF2. You can't actually make any new proteins. And so these host cells that this happens to essentially become zombies. They can't do anything because they can't make anything new. They eventually die, but before they die, you do all kinds of other crazy things. One interesting sort of biotechnological aspect of toxins is that if we go back and look at this structure, is that these binding domains and these active site domains are actually fairly modular. And so diphtheria toxin is actually an interesting one in that there's some engineering examples where people have replaced this A domain with other small enzymes that can survive basically exposure to acidic conditions, and you can basically get these sort of enzymatic payloads delivered into specific cells, sort of hitchhiking on toxins. So that's kind of one interesting way that, in terms of biotechnology, we can use toxins for our own purposes, because these things have been so exquisitely honed over evolutionary time to form these functions that these sort of enzymatic functions, we can essentially swap those out for things that we're interested in delivering into cells. Okay, so this is going to be one of the few toxins that I'm going to burrow down into like mechanism, because I think this is really interesting. And what they do is that elongation factor 2 has a conserved histidine in its active site. So histidines are interesting amino acids in that they're very sort of catalytically friendly, in that they're basically able to accept and donate electrons very well. They carry differential charge under different conditions. They can be neutrally charged, they can be positively charged. And so what ends up happening is that diphtheria toxin, requires that this histidine on EF2 be charged with a methionine residue, essentially. So acetyl methionine gets added on. But long story short, you form a specialized histidine, a post-translationally modified histidine EF2, and that this modified EF2 is actually the substrate for diphtheria toxin. And so we actually have a whole bunch of genes that have been sort of characterized as these sort of diphtheria toxin-generating kind of enzymes that we naturally perform this modification on on this particular histidine, or eukaryotes do in general. And this is part of what sort of drives the selective activity of this particular toxin on elongation factor 2. So elongation factor 2 is one of the few proteins that we have that gets this post-translational modification to appropriately interact with tRNAs. And so this is how one of the ways that diphtheria toxin selectively ADP ribosylates this particular histidine residue is because it is post-translationally modified by us. So, all right. So our first example. Are there any questions about this particular toxin?
What did you say the histidine is charged with what?
So
For the elongation.
So this particular histidine that's in this active kind of binding site of elongation factor 2, we charge this. So we use a substrate, acetyl methionine, which is it's one of the ways that that a lot of organisms post-translationally modify proteins, especially at histidines and lysines, with this particular chemical group that's on acetyl methionine. And so this part here is methionine that's been charged with an adenine nucleotide. And so what this does is that this is a really highly reactive intermediate compound, that there are a lot of what are known as radical SAM enzymes that add this essentially methionine without the sulfur onto things like histidine and arginine. And this is one of the important post-translational modifications that can happen to specific proteins in our cells, and there are some bacterial examples of these sort of modifications happening too. But importantly is that this particular diphtheria toxin, it's actually looking for this post-translationally modified histidine. So it doesn't necessarily work.
So once it's bound like to that, that's when it becomes a toxin.
Yeah. So this is what this toxin, or this ADP ribosylase enzyme portion of this toxin, this is what it's using as a substrate. And so this particular histidine, or this nitrogen here, needs to be basically activated by adding on this post-translational modification to histidine for this ADP ribosylation. All right. So, are there any other questions about this toxin? All right. So, I guess before we head into this one, let's take a quick break and go after a quicker question. I think hopefully should be a pretty straightforward time. And that superantigens activate T helper 1 cells independent of MHC2. Is this a true statement or a false statement? Yep.
I work for a full-time. Are you sure? Because I got an update. I know. It's just every time I pass my breath, I'm usually using my laptop.
Okay.
I swear I refreshed it like twice.
I don't. That's one of the headache
Yeah,
heartaches.
so what is that? For someone who's looking at an angle? It's like how.
All right. So, another 5 seconds or so. Make sure you get your answers in. All right. Let's take a peek. All right. So we've got a large number of B's and a small group of A's. So what am I asking here is when we think about superantigens, indeed they do activate T helper 1 cells. And the way that they do this, remember, is that they're forcing this interaction between the T cell receptor and CD4 and MHC2 to happen. It can just happen irrespective of what antigen is charged into MHC2, okay? And so this activation of T helper 1 cells via superantigens is indeed, it does absolutely require MHC2. So it requires it requires all of the normal pieces of a normal immune, or a normal T cell helper T cell activation event. It just happens regardless of whatever antigen is there, okay? So the answer here is false. So, all right. So we will quickly talk here about tetanus toxin. And it's produced by a gram-positive bacterium, Clostridium tetani, and is a well-controlled pathogen in terms of vaccination now. So many of us have received a tetanus vaccine. So if we basically step on a rusty nail, we're not going to get tetanus. And so the whole idea behind why this particular pathogen is associated with I stepped on a nail or I had some sort of puncture wound is that this bacterium is an obligate anaerobe and it's spore forming. And so unlike sort of open kind of lacerations on your hand, so if you just had a big cut, right? But that's still a reasonably aerobic environment. So it's at least micro aerobic. Now, if you have a deep puncture wound that, especially once deep in that wound, it has basically gotten clotted over with blood, or basically blood clots have formed deep in that wound. Now that is a deeply anaerobic environment that promotes sporulation of these Clostridium tetani spores. And basically, once they begin growing and they're in this anaerobic environment, they start secreting a neurotoxin of this AB variety that causes tetanus. And so, tetanus is characterized by spastic muscle paralysis. And so essentially this is your muscles flexing as hard as they possibly can. And this is an image drawn by Sir Charles Bell in the mid-18th, early 19th century. And basically just pictorially describing what someone racked with tetanus looks like. Basically all of the kind of large major muscles in your body are contracting and basically bends you into kind of a pretzel, more or less. And so Fortunately, we have very effective vaccines against this particular toxin. So while we might end up with very localized, controlled Clostridium tetani infections from puncture wounds in our daily lives, if we've been immunized against tetanus toxin, we're essentially able to inactivate this toxin as soon as it's produced by a local infection. And it's still a significant cause of infant mortality in the developing world. So this is one of the kind of global efforts of getting large populations throughout the world vaccinated for particularly nasty bacterial toxins. So I used to give the contrasting examples of tetanus toxin and botulism toxin. Important to know it's a single chain toxin, and it's actually, in terms of structure, not too disparate from our last example for diphtheria toxin. And so what's different about it is that it binds to gangliosides on inhibitory neurons. And so there are a number of different neurons in muscles that activate muscle contraction, and there are essentially other neurons that inhibit those activating neurons, and I'll kind of explain this in a second. This is another toxin that's delivered. The active domain is delivered into the neuron's cytoplasm by endosome acidification, and what's contrasting is that this active domain is actually a zinc-dependent protease. What this protease does, and I'll explain this in a second, is it cleaves a particular family of proteins, snare proteins, and this particularly important receptor VAMP. And what this does when this VAMP receptor gets cleaved is that these inhibitory neurons no longer secrete a neurotransmitter glycine. And so what's going on is that normally what we're doing when we're not contracting muscles is that these inhibitory neurons are secreting glycine. Now, glycine is bound by receptors on these sort of muscle activating neurons, that essentially what this does is stops secretion of acetylcholine by these activating neurons. So if we basically want to flex our muscles, these inhibitory neurons stop secreting as much glycine, and that then these activating neurons are able to start secreting acetylcholine, and that the muscle tissue that's responding to acetylcholine is going to contract. So what tetanus toxin does is that it basically blocks secretion of glycine so that these activating neurons are essentially always on, so that there's nothing ever turning them off. And so they're constantly secreting some amount of acetylcholine all the time, which builds up and essentially results in sustained muscle contraction. So what's going on with this particular toxin? And there's a lot of different things in this particular figure. And the thing that I like about the figure is that it's pointing out that botulism toxins, or that these clostridial toxins, or they kind of get grouped into being called botulinum toxins, is that they're very diverse, but they all basically kind of do the same thing. And that they're taken up into different types of neurons, because they're binding to these ganglioside receptors on the host surface of the neuron surface. These are taken up into vesicles, because these neuron cells are cells that actually like to form vesicles very readily. So this is taken up, and basically these different toxins do different. They basically are all proteases, but they all cleave different receptors from the same large family of snare receptors. And so depending on what they cleave, they're basically inhibiting different stages of neurotransmission. And so the example that I showed you is essentially disruption of secretion of glycine. Now, you can also imagine some of these same toxins inhibiting secretion of acetylcholine. And you can also imagine them inhibiting secretion of other neurotransmitter chemicals. So they basically are disrupting the normal queued up sets of events that are supposed to happen for neurotransmission by essentially cleaving, very specifically, different components of this neurotransmitter secretion pathway. And inhibitory neurons, activating neurons, and basically all kinds of other neurons. Okay, are there any questions? So who thinks these examples are boring?
Wait, you said, is this boring?
Is this boring?
Oh, no.
Okay, so we have one no.
No.
No? Okay. Is there a yes? It's okay if it's yes. I've taught this a bunch of times, so it's kind of no. The thing that is really crazy about this is just how different all of these toxins are. They all kind of have these sort of unified principles. And it's sort of this very modular thing that, like, you have a different binding domain that lets you interact with a different type of cell, and you have an active domain that's going to do something inside of those cells, right? And then what you'll see when you really start digging into a lot of different toxins is that this active domain has extraordinary specificity for things that are in particular types of cells. So there is this post-translationally modified histidine. That is what gets ABP ribosylated, for example, in the last example. In this case, these clostridial toxins, they like neurotoxins. They essentially like to preferentially cleave different snare proteins that are involved in neurotransmitter secretion. Okay. So we're going to talk about anthrax toxin, but before we do, I wanted to ask you guys. Oh yes.
I was wondering why we need to get a tetanus booster every 10 years.
So that's really an issue of antigenic number. And so it's basically how long, on average, most people maintain antigenic memory for that particular toxin. Yeah, good question. And so the booster is saying a nice round number of 10 years. So when we get a booster for it, 95% of this room might totally be fine and still have lots of antigenic memory against the tetanus toxin. But you're basically making sure that everyone's okay. So, yeah, good question. Yep.
What's the route of exposure for the infants? You said we see it from deep wounds. What's happening with infants that it's killing them at such a high rate?
What we'll see with a lot of these toxins is that you and I, we're all healthy, we have a robust immune system. Is a lot of these toxins are highly antigenic, so we'll develop adaptive immunity to them if we've seen them. And it's just kind of this idea that there's a lot more. There's a number of arguments, but there's a lot more volume for us. That we're generally better at clearing bacterial pathogens than small children, and that exposure to bacterial toxins in children is where these things are absolutely worst. So you and I got tetanus. I mean, if it was unchecked and we basically just had this festering wound in our foot for weeks, we might end up being that racked person with a severe case of tetanus. But for a small child, those disease symptoms happen. Good question. Yeah. Okay. So you guys had a good while to read this one. But pore-forming toxins, what are they going to need to do? Do they need to interact with the host cytoplasm, host membrane, host signaling pathways, host RNA polymerase, or host metabolic enzymes?
They're saying so that it goes from human trafficking. I just read this as a.
All right, let's try and get our final answers in. All right. So, all right, 100% of you said B. Great. So remember, spore-forming toxins, making holes, host cytoplasmic membrane. Okay? All right. Moving on. Anthrax. Not the heavy note. So, Bacillus anthracis is the source of anthrax toxin. It's a particularly nasty pathogen when you are exposed to it. But exposure is very, very rare. So it's a gram-positive rod. It's very closely related to the lab model bacterium, Bacillus subtilis. They both inhabit soil, and they're both spore-forming bacteria. So understanding a non-pathogenic bacillus has told us a lot about this particular bacillus does in its life cycle. But some of the important things that this particular other species that is not friendly to us is that it produces an AB toxin that causes a wide variety of symptoms ranging from skin lesions to acute systemic disease. So this can result in essentially really nasty necrotic lesions where you end up with Bacillus subtilis infection, or you can end up with essentially systemic problems that can be mediated by this particular toxin. Essentially all the sort of bio-terror stuff, bioweapons that have been developed around anthrax toxin are really aiming at eliciting this systemic response very rapidly. So if you ever want to read a really terrifying book, there's a book called Biohazard by Ken Alabak. So he was a chief bioweapons engineer in the old Soviet Union, defected in the late 80s, came to the US and essentially wrote this book that tells this terrifying tale of how he and his research group weaponized anthrax amongst other things. And so forms of anthrax, the actual disease, you can have cutaneous anthrax, and so that's, you were to basically type into Google, please don't do this now, type in and say, anthrax. One of the pictures you're probably going to see is some like big black lesion on someone's arm. It looks like their arm's going to fall off. So this is essentially cuts where you end up with a localized Bacillus anthracis infection. Where you start producing anthrax toxin, and you basically have this localized, really terrible response. You can get it by consuming food that's infected with anthrax. And so that's actually what happens to a lot of animals. So there's sort of these kind of these cursed fields in Europe and other places in the world where like if your animals go out and eat there, they're going to die. And what that is that those particular fields have Bacillus anthracis spores in them. Cows and other animals will eat them or will eat them. Eat soil that's contaminated with these spores, and then essentially these animals die from gastrointestinal anthrax infections. We can get these things. We can get them from eating the animals that have been infected with anthrax, and we can also inhale this because anthrax, essentially the bacterium forms spores, and if we inhale these spores and they germinate in our lungs, we end up with essentially a lung version of this cutaneous infection. So see a picture of the cutaneous infection. Imagine that in your lungs. And, that is why this is particularly nasty. So, Bacillus anthracis has been used or developed as a biological weapon a number of times in history. Here's a number of examples. You can essentially kind of look up at your leisure and see where this has been used for the ultimately wrong kind of purposes, ranging all through the 20th century and the early part of the 20th century, 21st century. So the characteristics of anthrax is that it is a multi-subunit toxin. And so this is now our first example of something else, right? It is a multi-subunit toxin that contains two different active subunits. So essentially the binding part of this toxin is the same, but there's two different active subunits. And so it binds to a particular receptor, uncreatedly called anthrax toxin receptor, on host cells. It has, at least the last time I checked, whether or not we know what this receptor does. I'm going to say that it is one of these, like, we don't really know what it does, but anthrax toxin likes to bind to it. And it's delivered to the cytoplasm, again, by endosomal acidification. I promise we'll get in another example. And so one of these active subunits, once delivered into the cytoplasm, is called lethal factor. And so this is a zinc-dependent protease that cleaves MAP kinase proteins. And so MAP kinase proteins, remember these are proteins that are involved in essentially phosphosignaling cascades that shape how cells sense and respond to a large number of things. So there's a lot of different MAP kinase cascades in eukaryotic cells, and that what these lethal factors are going around and doing is cutting up all of these different MAP kinase proteins, essentially inactivating all kinds of signaling cascades in host cells. The other is this edema factor, or EF, and this is an adenylate cyclase. And what this does is causes inappropriate production of a signaling molecule, cyclic AMP. So those of you that, if you think back to where you may have heard of cyclic AMP before in microbiology, is talking about the lac operon, and that you have this sort of starvation signal. Turns out that we also use cyclic nucleotide signaling. It's a fairly conserved thing throughout all living things. And so if you inappropriately make cyclic AMP at the wrong time, cells are going to inappropriately respond to this and do things that they wouldn't normally do. And so one of the things that happens with adenylate cyclase and inappropriate cyclic AMP generation is basically that cells begin pumping out a lot of water, and so tissues become basically engorged with water, and so you retain a lot of water. And a whole bunch of cells that are also infected with the same toxin are going to start dying, and you basically end up with just an environment ripe for a really nasty necrotic infection. So in either case, this pictorially what's going on here is that there is this B domain that binds to, that essentially interacts with anthrax toxin receptor, and that this binding subunit interacts with either the lethal factor or the edema factor. In either case, endosomal acidification leads to entry of these different active subunits into the host cytoplasm. So either you're destroying MAP kinase proteins that will ultimately lead to cell death, or that these cells begin overproducing cyclic AMP that results in expulsion of water out into the surrounding tissue, and so basically these tissues become the intracellular space between all the cells in the tissue get engorged with water. So are there any questions about this particular toxin? All right, so we've seen an ADP ribosylase, we've seen a couple of proteases, and we've seen some single subunit, basically single chain toxin, AB toxins. We've seen now an example of sort of multiple subunit toxins. And what I want to take you through next is cholera toxin. And cholera toxin is of a particular variety of AB toxins. So this is this ANB toxin, or AB5 in this case toxin. And this is a toxin that causes acute diarrhea and dehydration. So it's actually going to do some things akin to what that edema factor does in anthrax toxin, and that what it's going to do at the end of everything I'm going to tell you is that it results in overproduction of cyclic AMP, and overproduction of cyclic AMP in host cells results in expulsion of water and chloride anions and other things that basically end up out in the extracellular environment. And in this case, the extracellular environment is the lumen of your intestines, and not, subcutaneous tissue in your arm, for example. And what this does is that it basically causes horrible watery diarrhea. And so it's a multi-subunit toxin, and it binds to GM1 gangliosides on intestinal cells. And so this is another example of these gangliosides. They're a particular type of lipid, and that this is a frequent site of toxin. These binding interactions of toxins are often with different types of gangliosides that are on different types of host cells. In this case, these GM1 gangliosides, these are found on intestinal epithelial cells. So, unlike our previous examples, this is our first example where this toxin is delivered to the cytoplasm via retrograde transport. And so so this A subunit, we're going to see that it performs more or less the exact same chemistry that we saw with diphtheria toxin. It's just going to do it on a different protein, and that protein ribosylation, ADP ribosylation of this protein, is going to result in overproduction of cyclic AMP. So, how does this work? So again, everything's going to come in as an endosome. And so in the examples that we talked about before, that endosome will get acidified, and that is an activating trigger for this toxin. Whereas this toxin, cholera toxin, ends up being transported through retrograde transporting into the ER. And basically this active subunit, after some processing in the ER, is then secreted back out into the cytoplasm, where it goes and ADP ribosylates a family of proteins that is involved in regulating our own native adenylate cyclase activity. So they essentially are normally inhibiting adenylate cyclase activity. So essentially we're taking the brakes, this toxin takes the brakes off of adenylate cyclase. So before, what we saw was that edema factor itself is an adenylate cyclase. And so the enzymatic activity of that toxin is to overproduce cyclic AMP. And so overproduction of cyclic AMP is going to result in release of chloride anions, also a lot of water. And what we see with cholera toxin is the same end result, that it is perturbing the intracellular pool of cyclic AMP, but it's doing it via a very divergent mechanism. So it's essentially going to take the brakes off of our own adenylate cyclase and allow it to produce a lot of cyclic AMP. So what normally happens is that there are these G proteins that are important for signaling and essentially controlling when and how much cyclic AMP is produced by our cells. So under normal healthy conditions, our cells are not really producing a lot of cyclic AMP. But if they're stressed, either nutritionally, or there's oxygen depletion, all kinds of things will cause these G proteins to essentially not get phosphorylated. And essentially the brakes are then taken off of adenylate cyclase. We start producing a whole bunch of cyclic AMP, and there's a bunch of downstream signaling that happens as a result. So what cholera toxin does is it ADP ribosylates this G protein, G signaling protein, and essentially makes it inactive. So no longer can we phosphorylate or unphosphorylate this protein and have it control when adenylate cyclase is active. This toxin, through ADP ribosylation, inactivates the signaling protein, taking the brakes off of cyclic adenylate cyclase. Our cells begin overproducing cyclic AMP and start basically secreting a whole bunch of water, chloride anions, and carbonate. Yep.
Quick question, can we go back to the picture before that? So when we're producing the Cl ion, are we producing water, or is water moving to the lumen to neutralize
So
the ion?
it's moving along with
Okay.
chloride. Yep. And also with carbonate. So essentially what these toxins are doing is triggering these cells into thinking that they're starving. And they start doing basically excessively what our cells normally do when they're starving, which is secretion of water and carbonate, all kinds of stuff. So what I'm showing in this slide is kind of the progression of what begins to happen as a result of what's happening to intestinal epithelial cells when they're exposed to cholera toxin. But again, there's binding, we take this stuff up. This active subunit ends up out in the cytoplasm, and basically this exacerbates, it takes the brakes off of any control we have over production of cyclic AMP in these cells. Overproduction of cyclic AMP results in secretion of chloride anions, carbonate anions, and basically disrupts sodium transport across these epithelial cells. And one of the net results of this is both active and passive transport of water to basically charge methods. What's happened, basically moving all these anions and not allowing cation transport. Okay. So are there any questions so far? So the big molecular connection here is that in both edema factor and cholera toxin, cyclic AMP signaling is a central thing to our cells in our body. And so if you target a particular family of cells and you disrupt that signaling, you get all kinds of crazy things to happen to these tissues. In this case, and in the same case with edema factor, is that these cells start pumping water, I shouldn't say pumping, but water starts leaving one set of tissues into the extracellular space of another, and you essentially can end up with edema, which is basically water retention, uncontrolled water retention in tissues. And in the case of cholera, this same uncontrolled water movement is just into the lumen of your intestine instead of kind of tissue in your arm. And I'll leave you all kind of to imagine what your your bowels being filled with water that's going to happen as a result of that, and that this is going to be sustained over days of you doing this. And so if you do have cholera, one of the things that they're going to do is hook you up to an IV, make sure you're well hydrated, and you're going to lay on a cot that has the hole cut out in the middle, and they're going to put a bucket underneath you, and you're going to lay there in agony for 5 to 7 days. So this is the thing that, you know, dealing with clearing cholera is that it is generally a self-clearing pathogen. It's just that the intense water loss that happens as a result of the activity of this toxin really makes this extremely dangerous for us, and again, really for the children. Right? Uncontrolled water loss for children, you know, we're physically larger, we're a bit more robust than small kids. So intense water loss in children is really, the prognosis is good, right? So, and we'll talk a little bit later about why the heck is there not an awesome cholera toxin vaccine. And so we'll get to that in a few lectures. Okay, so the last toxin that I'm going to take you through is one that we're actually going to talk about on Tuesday, or at least a derivative of this same toxin. And this is a toxin known as cytolethal distending toxin. And so it's a tripartite toxin, and it has a number of different arrangements in a number of different organisms. The example we're going to talk about next time, or in our next, yes, on Tuesday, is A1B1B25. So it takes a really strange conformation. And it's classified as an AB2 toxin because it has essentially 2 binding domains and 1 active domain. And binds, where do these things bind? They bind to cholesterol-rich rafts and host cell membranes. And so this is a pretty generic toxin in what it will interact with, because lots and lots of our cells have cholesterol-rich rafts. And so it contains an active subunit that actually has 2 strikingly distinct activities. One is that it is a DNA nuclease, and it is also a phosphatidylinositol phosphatase. And so I will try and be clear about what these 2 things are in just a second. But what is phosphatidylinositol? Anyone know? Well, phosphatidylinositol is a type of glycolipid that is predominantly found in eukaryotes. So it's a normal phospholipid that has an inositol head group. So if you go back to one of our early lectures where I showed you what lipids can look like, they can have different head groups. And inositol is one that is frequently found, well, it's predominant in eukaryotes. So it's a very abundant lipid in eukaryotic membranes. So phosphatidylinositol, phosphatidylcholine, among others, phosphatidylethanolamine. Phosphatidylinositol is something we have, and it's actually a really important signaling lipid, and I'll get to that in just a second. So what the active subunit does when it is in the cytoplasm and it's doing its dirty work is that it causes cell cycle arrest at G2, the transition from G2 to M. So basically, gap 2 to mitosis in host cells. So essentially, actively growing and dividing cells, they get locked into this state. And we'll see that in a second. And again, it contains the binding subunits, and it is delivered to the cytoplasm by a retrograde transport. And so what we'll see is when we see these AB5 toxins, that these almost always are delivered, that these are the retrograde transport toxins. A lot of the other toxins, they are vacuole, sort of like vesicle acidification toxins, whereas these AB5 arrangements that some of you might see with some of your pathogens, that these are almost always delivered through retrograde transport. So it's a preserved kind of mechanism for this kind of structural toxins. So when I'm talking about arrest, and where this happens is that if we think about the normal cell cycle, there's start in a freshly minted, freshly divided cell in G1, basically goes through G2, and then we're going into, essentially we're going to start making a new cell, and essentially where things get arrested, as we're going to see in our paper on Tuesday, is that these cells get arrested here with this this stage where essentially normally we'd be going through segregating chromosomes into two daughter cells that basically this gets interrupted. How does it get interrupted? Well, this toxin gets delivered by retrograde transport, and the active subunit, if it gets secreted to the cytoplasm, basically goes to phosphatidylinositol lipids, and depending on the phosphorylation state of these phosphatidylinositol lipids, because it's a carbohydrate head group, and it can carry up to, I think, 5 phosphates, but it's usually 1, 2, or 3. And these triphosphate phosphatidylinositols, they are something that is a signal to a protein AKT. AKT gets phosphorylated. When these things are phosphorylated, and this phosphorylated form of AKT basically promotes cell division. So this is one of these sort of membrane signals for like, yeah, now's a good time to promote cell division. Whereas what this B unit does on the cytoplasm is dephosphorylates these triphosphatidylinositol lipids, and essentially interrupts this normal signaling event so that AKT never gets phosphorylated, and so that cells don't, that cell division is no longer promoted. The other thing that they do is that sometimes this toxin, instead of getting secreted out into the cytoplasm, gets secreted into the nucleus. And when it's in the nucleus, it cleaves DNA, basically destroys the host chromosome. You have a massive DNA damage response that ultimately, the coordinated effort of both arms of what this toxin does, basically arrests cells in G2M. And so essentially you're no longer able to proceed to grow and divide. So what's interesting is that you might say like, well, maybe one of these is more important than the other. And what it turns out is if you disrupt these corresponding activities in this toxin, is that both seem to be really, really important for causing this arrest at G2M, but we don't really understand. So in tissue culture, we know exactly what happens with this toxin, but exactly what it's doing in the host and why, what it's actually, how it's eliciting a disease presentation is not really fully understood with this toxin. So it's produced by a large number of gram-negative pathogens. So we're going to see Salmonella typhi produces this. And this is an important part of basically causing typhoid fever for this particular Salmonella derivative, or Cerevar. It's also produced by some Haemophila influenzae. It's produced by some toxigenic E. coli. It's produced by some Shigella species. It's produced by a lot of different gastrointestinal pathogens and basically systemic pathogens, and basically does this by cleaving DNA. So I will leave you today to think about what fills in these boxes. So how are these toxins arranged? What do they do? And what are they targeting? Okay. So. Are the instructions for the assignment for Tuesday, are they clear? So So the discussion, you're going to post 3 questions. And then after you post your questions, you should be able to see everyone else's questions. So what you're going to need to do is to go find 2 questions from 2 separate peers in the class that have not yet been answered. And answer 2 questions. Okay? So you get basically 6 points for your questions and 4 points for your responses.
Don't be restating the question again.